Welcome, colleagues, researchers and knowledge seekers alike. This edition, entitled Scientific Limitations and Research Gap, SLRG Master 01, is presented by Brian Geniuson, Research Systems Architect and Independent Research Scientist, Natural Scientifics. The presentation introduces a systems-level framework designed to evaluate the structural stability of biomedical evidence. It is not a therapeutic claim platform. It is an evidence architecture audit model. Modern biomedical research operates across multiple layers. These include molecular biology, preclinical experimentation, statistical inference, clinical trial design, epidemiology, and real-world implementation. Each layer introduces distinct forms of vulnerability. When these vulnerabilities accumulate, translational stability weakens. The Scientific Limitations and Research Gaps framework organizes these weaknesses into 6 interacting domains. Preclinical science gaps, statistical and analytical gaps, clinical trial design gaps, translational and implementation gaps, epidemiology and funding bias gaps, mechanistic biology gaps. These domains form a fragility network. They do not compete. They compound. Preclinical science often relies on simplified in vitro systems, species-specific physiology, and dosing paradigms that may not reflect human pharmacokinetics. Aging models and comorbidity modeling are frequently absent. As a result, mechanistic signals may appear strong in controlled environments, yet weaken in human translation. Statistical fragility emerges from multiple comparison inflation, selective endpoint reporting, insufficient pre-registration, and model overfitting. Without replication, inference stability declines. Clinical trial design limitations include underpowered samples, surrogate endpoint dependence, short follow-up duration, and limited population stratification. These factors reduce reproducibility and external validity. Even when clinical efficacy is observed, real-world implementation may fail. Bioavailability variability, pharmacogenetic heterogeneity, polypharmacy interactions, adherence constraints, and cost access barriers create translational discontinuity. At the population level, epidemiologic inference is vulnerable to confounding variables, reverse causation, self-report bias, and funding influence. Mechanistic biology gaps arise when complex biological systems are reduced to single pathways. Correlation may be mistaken for causation. Tissue specificity, temporal dynamics, and multiomics integration are often underrepresented. To operationalize these vulnerabilities, the framework introduces the Evidence Fragility Index. This index evaluates research stability across 4 non-overlapping dimensions: mechanistic certainty, preclinical robustness, clinical reproducibility, translational readiness. This structure allows formal identification of evidentiary ceilings and fracture points. The purpose of this master series is not to diminish scientific discovery. It is to reinforce structural credibility. By formalizing scientific humility and methodological transparency, the Scientific Limitations and Research Gaps framework strengthens translational accountability across biomedical research. Full scientific literature references are available in the video description. This content is provided for educational and scientific discussion only. It does not constitute medical advice and should not replace consultation with qualified healthcare professionals.